Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway. Okay, right. So we are currently in the process of discussing what a phosphoglycerate lipid is. Specifically, we want to look at what phosphatidyl inositol is. Okay, so we've seen that a phosphoglycerate lipid is produced from this molecule phosphatidic acid by adding on an alcohol group containing molecule onto the phosphate group of the phosphatidic acid molecule. Okay, and that's how we create a more general phosphoglycerate lipid. Okay, so the example of a phosphoglycerate lipid that we're extremely interested in is phosphatidyl inositol. And in phosphatidyl inositol, you're going to add on an inositol molecule onto that phosphatidic acid molecule. Okay, so let's now discuss inositol. So inositol is the old name for a molecule which would now be called cyclohexane 1, 2, 3, 4, 5, 6 hexol. Okay, so cyclohexane 1, 2, 3, 4, 5, 6, and then finally hexol. So let me just bring this over a little bit. Hexol here. Okay, and although that certainly is much more of a mouthful than inositol, it's a useful name because it tells us exactly what we're dealing with. Cyclohexane tells us that we're dealing with a 6-membered carbon ring. Okay, where all of the bonds between the carbon atoms are single bonds. Okay, so if we draw a skeletal formula for it, it'll be like so. Remember, in skeletal formula, you don't show carbon atoms, they're implicitly shown by corners. So these 6 corners here are implicitly showing carbon atoms. So this is cyclohexane here. Okay, now each one of those carbons now needs another 2 bonds coming from it. Okay, one of these bonds will be to an alcohol group, since absolutely every single one of the 6 carbons is going to have an alcohol group coming off it. Okay, and the other bond is going to be to a hydrogen atom. Okay, so every carbon has both an alcohol group coming off it and a hydrogen coming off it. Now you might think, oh, how beautifully simple. However, there's a subtlety here that is not so simple. And that is that there are going to be multiple optical isomers of this. Okay, so let me explain this. So, if you imagine that the 6 carbons of this 6-membered cyclohexane ring here are sitting within the plane of the piece of paper. Okay, so imagine that we've arranged them so that they're nicely sitting in the plane of the piece of paper. Then the other bonds that come off each of these carbon atoms, one is going to be coming off at an angle like this, okay? And one is going to be going into the page away from us at an angle like this, okay? So either the alcohol group can be the one coming out of the page towards us and the hydrogen can go into the page away from us, or it can be the other way around. The hydrogen can come out of the page towards us and the alcohol group can go into the page away from us, okay? So there are different ways of arranging these 6 alcohol groups that are coming off these 6 carbons here. Okay, and this gives rise to multiple optical isomers of inositol. In fact, there are actually 9 different optical isomers of inositol. Now, luckily, there's only one major optical isomer of inositol that is actually used within cells, and this is called myo-inositol. So what I'm now going to show you is the optical isomerism of myo-inositol. So let's start with this carbon here. Okay, so I'm not going to show the hydrogen atom coming off. All I'm going to show is the alcohol group coming off. Okay, because remember we're drawing a skeletal formula so that you don't need to show hydrogen atoms coming off carbon atoms. Okay, so the alcohol group here is going to come out of the page towards us. And the way you show this is by one of these bonds that gets thicker towards the end. Okay, so here is the alcohol group that's coming out of the page towards us. Again, off this next carbon down here, we're also going to have an alcohol group that comes out of the page towards us. Okay, like so. Then off this one, we're also going to have an alcohol group that comes out of the page towards us. So another one of these bonds that gets thicker towards the edge. And then finally, on this carbon, we're actually going to see one that goes back into the page away from us. So the way I'll show that is by a dashed line. Excuse me, by a dashed line like so. Okay, so the alcohol group is this time going into the page away from us, which means that the hydrogen will be coming out of the page towards us. Okay, then on this next one, the alcohol group will once again be coming out of the page towards us, and therefore the hydrogen will be going into the page away from us. And finally, off this 6th carbon here, the alcohol group will be going into the page away from us, and then the hydrogen will be coming out of the page towards us. So this is the structure then of myo-inositol, this specific optical isomer of inositol. Okay, right. So, if you say have an inositol molecule and you want to work out whether it's a myo-inositol molecule, you need to try and rotate it around and try and superimpose it on this picture I have here. If you can do that, then the carbon atoms of that myo-inositol ring have a specific way that they need to be labelled. Okay, so this one here, this is always named the 1st carbon. This one here, the 2nd, the 3rd, the 4th, the 5th, and the 6th. Okay, and this is really the reason that I am stressing this optical isomerism of inositol so much, because there is this set naming system for the carbons of this myo-inositol ring. Okay, so if you've got an inositol molecule, you need to try and superimpose it on this image. If you can do that, then you name up the carbons of your inositol ring according to which carbon they are sitting over once you've superimposed that ring on this drawing here. Okay, right. And that's going to become important when we come to the naming of phosphoinositides. So what we're now going to do is we're going to take our phosphatidic acid molecule, okay, here, and we're going to attach onto the phosphate group of the phosphatidic acid this inositol ring, and specifically we're going to attach this alcohol group that comes off the 1st carbon of the myo-inositol ring. Okay, so here is our inositol ring here, which I'm just going to now show as a hexagon. Okay, so let's colour this diagram in. So here we have the long chain carboxylic acids, which remember are esterified to the 1st and 2nd carbons alcohol groups of the glycerol molecule. Here is our glycerol molecule in green here. Okay, then coming off the alcohol group that comes off the 3rd carbon of the glycerol molecule, we have our phosphate group, and now we've attached our phosphate group to the alcohol group that comes off the 1st carbon of our myo-inositol ring. Okay, right, so this molecule that we have now created is a phosphoglycerate and it's a phosphoglycerate called phosphatidyl inositol. Now this is a running theme for all of the phosphoglycerate. They begin with this prefix, okay, phosphatidyl. Now what phosphatidyl means is I contain a phosphatidic acid molecule here, which I'll circle in red. Okay, so that bit is present in all of the names of the phosphoglycerate because they all contain the phosphatidic acid as their backbone, if you like. Okay, and then the next portion of the name just tells you what you've got stuck onto the phosphatidic acid, which in this case is an inositol ring, so that's phosphatidyl inositol. Okay, so phosphatidyl inositol is often abbreviated to PI. Okay, right. Now, you do have a little bit of phosphatidyl inositol within your phospholipid bilayers of your cell membranes. Okay, now what we want to discuss is what a phosphoinositide is. Okay, so phosphoinositides are going to be derivatives of phosphatidyl inositol. A phosphoinositide is a phosphatidyl inositol molecule that has been phosphorylated on the alcohol groups of these other 5 carbons here. Okay, so if you take phosphatidyl inositol and add at least one phosphate group onto the alcohol groups of these other 5 carbons of the inositol ring, then that creates you what is known as a phosphoinositide. So let me give you some examples. We could stick a phosphate group onto the alcohol group that comes off this 4th carbon here. Okay, again via a phosphoester link here, like so. And that would now be a phosphoinositide. The name of this phosphoinositide would be phosphatidyl inositol. Okay, we've always got phosphatidyl inositol. Okay, and now it's got a phosphate group coming off the 4th carbon of the inositol ring. Okay, so this would be phosphatidylinositol 4-phosphate. Okay, and for short, phosphatidylinositol 4-phosphate can be abbreviated to PI for phosphatidylinositol, and then 4, because we've got a phosphate group coming off the 4th carbon, and then P for phosphate. Okay, so PI4P stands for phosphatidylinositol 4-phosphate. Okay, and it's an example of a phosphoinositide. Another very notable example of a phosphoinositide is where you phosphorylate not only the 4th carbon's alcohol group, but also the 5th carbon's alcohol group. Okay, like so. So we're now phosphorylating this alcohol group up here. Okay, now this molecule then is going to be called phosphatidylinositol 4,5-bisphosphate this time, because we've got 2 phosphate groups coming off the inositol ring. Okay, well, we've got 3 including this one, but 2 accessory phosphate groups coming off the phosphatidylinositol molecule, which are coming off the 4th and the 5th carbons of the inositol ring. So this is phosphatidylinositol 4,5-bisphosphate, and this is why I wanted to stress the importance of understanding that this is a specific optical isomer of inositol, because if I had just drawn this picture, you might be wondering, well, why is that not called phosphatidylinositol 3,4-bisphosphate? Why is this not the 3rd carbon? Okay, well, you now understand that this 5th carbon is fundamentally different from the 3rd carbon. The 5th carbon is sitting between 2 carbons, the 4th and the 6th ones, which both have their alcohol groups going off in the opposite direction to the direction that the 5th carbon itself has its alcohol group going off in. Okay, so for instance, the 5th carbon's alcohol group comes out of the page, whilst 4th and 6th, the 2 that surround it, go into the page. If we now look at the 3rd carbon, that's not the case. In the case of the 3rd carbon, it's sitting between 2 carbons, one of which has its alcohol group going in the opposite direction to its alcohol group, but the other has the alcohol group going in the same direction as the direction which it has its alcohol group going in. So there is a subtle difference between the 3rd and the 5th carbons. So this comes off the 5th carbon, not the 3rd carbon. So that's why that's not called phosphatidylinositol 3,4-bisphosphate. 3,4 would be very different from 4,5. Okay, now, for short, phosphatidylinositol 4,5-bisphosphate is abbreviated to PI, and then we put brackets 4,5, and then P2, like so. However, you will often see PI45P2 abbreviated further. Okay, it's often abbreviated to PIP2. Okay, just to PIP2. And this does not tell you specifically which carbons of the inositol ring that these 2 phosphate groups come off. However, if you see this and it doesn't specify which carbons the 2 phosphate groups come off, you can assume they're coming off the 4th and the 5th. Okay, if they're not coming off the 4th and the 5th, no one would ever write PIP2. They'd have to specify that they were coming off different carbons. So for instance, if instead it was PI3,4-bisphosphate, they would most definitely specify PI3,4P2. Okay, they would never abbreviate PI3,4P2 down to PIP2. The only one that you can get away with reducing it down to this for is PI45P2, because it's so important in cell signaling. This is the most important of all of the phosphoinositides as far as cell signaling is concerned. Okay, so that one can be abbreviated further to PIP2. So those are 2 nice examples of phosphoinositides. Okay, right, so let's now go back to discussing these PI3 kinase enzymes, which I introduced so long ago. So these are phosphoinositide 3 kinase enzymes. Now we are in a position to understand what that actually means. These are enzymes which are going to add phosphate groups onto the alcohol group that comes off the 3rd carbon here. They are going to stick phosphate groups onto this carbon's alcohol group. Okay, that's what these enzymes do. They phosphorylate phosphoinositides on the 3rd carbon of the inositol ring. Okay, now, there are, broadly speaking, 3 different families of phosphoinositide-free kinase enzymes. Okay, so there are type 1 phosphoinositide-free kinase enzymes, there is then type 2, and then finally type 3. Okay, now, we are going to focus completely on type 1. Okay, they're the ones which are going to be important in the PI3 kinase AKT mTOR pathway. And if people don't specify which type they're talking about, you can assume they're talking about type 1 PI3 kinases. Okay, but just for a complete picture, I'm going to tell you the difference between type 1, type 2, and type 3 PI3 kinase enzymes. Okay, so we'll begin with the most important, type 1 PI3 kinases. These have as their substrate, okay, phosphatidyl inositol 4,5-bisphosphate, PI45P2, or just PIP2. Okay, so that's how these differ. They differ on which phosphoinositides they're actually going to add the phosphate group onto. Okay, so type 1 phosphoinositide-free kinase enzymes then are going to have as their substrate PIP2 molecules. Okay, so let's draw this out once again. So here is our inositol ring with the phosphate groups coming off the 4th and the 5th carbons of that inositol ring. So let's just colour this in. Here is glycerol in green here. Here are the 2 long-chain carboxylic acids in orange. Here are the phosphate groups, which we'll have in pink here. And then here we have our inositol ring in blue here. Okay, right, so that's the substrate. Now what type 1 PI3 kinases are going to do is they're going to phosphorylate the alcohol group that comes off the 3rd carbon of the inositol ring, this alcohol group here, and that's going to create us a new molecule, which is now going to have 3 phosphate groups coming off the phosphatidyl inositol molecule. Okay, so here once again is our inositol ring, and we now have phosphate groups coming off the 5th, the 4th, and now the 3rd carbons of the inositol ring. Okay, so here is our inositol ring. Here are our long-chain carboxylic acids in orange. Okay, here's our glycerol molecule in green, and now just to colour in the phosphate groups in purple here. Okay, so what's this molecule that we've created here now called? Well, firstly, it's going to start off with phosphatidyl inositol, as always. It can't not have that portion. Okay, so phosphatidyl inositol, if I can squeeze it in there. Excellent. And now we've got phosphate groups coming off the 3rd, the 4th, and the 5th carbon. So it's going to be 3, 4, 5, and then trisphosphate. Okay. And for short, phosphatidyl inositol 3,4,5-trisphosphate is often abbreviated to PI for phosphatidyl inositol. Then you brackets 3,4,5, okay, and then on the end you have P for phosphate, and then you've got 3 of them, so it's P3. So PI 3,4,5 P3 is the abbreviation for phosphatidyl inositol 3,4,5-trisphosphate. And again, this can be abbreviated, and this is the only one that you can get away with abbreviating down to PIP3. Okay, so if people just write PIP3 like this without telling you which phosphate groups, which carbons the phosphate groups are coming off, okay, you can assume that they are coming off the 3rd, the 4th, and the 5th carbon. Okay, this is the main PIP3. Okay, so we have converted then PIP2, PIP2, into PIP3, PIP3. Okay, right, so that's what type 1 phosphoinositide-free kinases do, and those are the ones that are going to be important in our pathway, so we'll come back to these. Just for a complete picture, I want to tell you also about type 2 and type 3 PI3 kinase enzymes. So let's start with type 2 next. So type 2 have a different substrate to type 1. Okay, so once again we'll start off with our phosphatidyl inositol molecule. Now the substrate for type 2 PI3 kinases is phosphatidyl inositol 4-phosphate. Okay, so we discussed this earlier when we were discussing examples of phosphoinositides. Okay, so this is phosphatidyl inositol 4-phosphate, PI4P. Okay, right, so let's colour this in. So here we have our long-chain carboxylic acids in orange here. Here we have our glycerol molecule in green. Here we have our phosphate groups in pink here. And then we have our inositol ring in blue here. Okay, now what type 2 PI3 kinase enzymes are going to do is they're going to phosphorylate the third, the alcohol group that comes off the third carbon of the PI4P molecule. Okay, and then what we're going to produce is this other phosphoinositide which has 2 phosphate groups coming off the inositol ring. Okay, we're going to produce PI3K. Okay, and this one you cannot get away with abbreviating this to PIP2. Okay, you have to state exactly which carbons of the phosphatidylinositol molecule the phosphate groups are coming off. Okay, so here is our inositol ring in blue here. Here is our glycerol molecule in green. Here are our long-chain carboxylic acids, the sterified to the alcohol groups coming off the first and second carbons of the glycerol molecule. Okay, so this now is phosphatidylinositol, PI, and now we've got 3. So we've got a phosphate group coming off the alcohol group of the third carbon, and we've also got a phosphate group coming off the alcohol group of the fourth carbon, so PI3K. Okay, and you cannot, as I say, abbreviate that down to PIP2. This is the only one you abbreviate to that. Okay, right. Finally, type 3 PI3 kinases, they work on just phosphatidylinositol itself. Okay, so here is the phosphatidic acid, and here is our myoinositol ring attached on. Okay, so let's colour this in. Here is our phosphate group in purple. Here is our inositol ring in blue. Okay, here's our glycerol molecule in green. And here are long-chain carboxylic acids attached to the alcohol groups that come off the first and second carbons of the glycerol molecule. Okay, and type 3 PI3 kinase enzymes are going to phosphorylate the third carbon's alcohol group of the inositol ring. Okay, so what you're now going to produce then is phosphatidylinositol with a phosphate group coming off the alcohol group of that third carbon, which is of course phosphatidylinositol 3-phosphate, PI3P. Okay, right, so let's colour this in. Here's our glycerol molecule in green. Here are our long-chain carboxylic acids in orange. Here are our phosphate groups in purple, and then we'll have the inositol ring in blue. Okay, so those are the 3 major types of PI3 kinase enzymes, and they differ, as I say, in their substrate specificities. Okay, so we are going to focus entirely on type 1 PI3 kinase enzymes, which are going to phosphorylate phosphatidylinositol 4,5-bisphosphate molecules, PIP2 molecules, into phosphatidylinositol 3,4,5 trisphosphate molecules, PI3,4,5P3 molecules. Okay, right. So, we'll call it there for this video, and in the next video what we'll do is we'll move on and have a look at the different type 1 PI3 kinase enzymes, because unfortunately there is not just one type 1 PI3 kinase enzyme, there are many different type 1 PI3 kinase enzymes.